Awesome.
So Matthew Shinkle is from the University of Nevada, Reno, and he is a PhD student, and he is going to tell us about body part and location selectivity surrounding human MT plus or human MT in bold responses to naturalistic stimuli. So please, Matthew, take it away.
Awesome. Thank you. You can see my screen fine? All right. So thank you, everybody, for coming. We actually spend a lot of time in our everyday lives looking at bodies, and this is because bodies can tell us a lot of things. Everyone from stage actors to advertisers to animators know that you can convey a lot about someone's intentions and communication and emotions just from the positions of their body parts. And so this raises the question of where bodies and body parts and their positions are represented in the brain. There are a few regions of cortex that are well known to be selective to bodies and body parts. These include the fusiform face and body areas, the occipital face area, and the extrastriate body area. But there have been a few papers in the last decade that have added nuance to this description. For instance, Orlov, Makin, and Zahari in 2010 showed participants images of isolated body parts versus control images and found selectivity to not just bodies, but specific body parts, forming this sort of patchwork across a large portion of lateral occipitotemporal cortex. Weiner and Grossbecker in 2011 did a similar study, but found that body selectivity was generally circumscribed to a region around hMT, a region of cortex known to be selective to motion. And they found that body selectivity could generally be described as 3 distinct clusters with selectivity to different body parts and visual field locations. Ferry, Kolster, Zastroff, and Orban in 2013 used maps of retinotopy and motion selectivity to determine the degree of overlap between motion and body selectivity around hMT, finding a large degree of overlap in this region. And so this raised a few questions that we were interested in investigating. We wanted to see whether we could predict bold responses to naturalistic stimuli based on body information. We wanted to determine the degree of overlap between body and motion selectivity, specifically around hMT. And we wanted to see whether body selectivity around hMT could actually be described as multiple body selective regions with different tuning to different body parts and visual field locations. So to address this question, we used 3 sets of naturalistic stimuli: a set of static natural images, a series of dynamic naturalistic movie clips, and a set of computer-rendered scenes with a camera smoothly panning through the environment. The fMRI data in response to these stimuli was collected for a number of previous studies, but for 3 of our participants, we had data for all 3 of these sets of stimuli, resulting in over 9.5 hours of richly varied naturalistic stimuli that are closer to visual experience than stimuli used in previous studies and conventional localizers. In order to extract body information from our stimuli, we used OpenPose, which is a deep neural network that extracts the locations of body key points from images or movie frames. And so a key point could be, for example, an elbow or a wrist. And then we use these key point locations in order to infer the locations of larger body parts in our stimuli, specifically the locations of faces, trunks, arms, hands, legs, and feet. In order to characterize the extent to which voxels are selective to these features, we employed a voxel-wise modeling approach. And this involved fitting a separate model for each participant and for each data set. To do this, we split our stimuli into training and testing stimuli. We used the body features and bold responses of a single subject in order to fit model weights using ridge regression. And here there is a model weight relating each voxel in a participant's brain to each one of our training features. We then use testing body features, so features from our testing stimuli, and multiply these by our model weights in order to predict bold responses at each voxel in the brain. We then computed the correlation between our predicted and observed bold responses at each voxel in order to generate a map of how well we can predict each voxel in the participant's brain just knowing these body features. So what this resulted in for each participant was 3 of these maps, 1 for each experiment, of prediction accuracy at each voxel. But because we wanted to combine data from all 3 of our data sets into our analyses, what we actually did was then average these flat maps into a single average flat map for each participant. And so here we have the average prediction accuracy across data sets for one of our participants using our body model. And so as you can see in regions such as FFA and OFA, we see fairly high prediction in voxels, which makes sense given that our model includes face information among other body parts. Now, if we look at this region around hMT, we see very poor prediction accuracy toward the center of hMT, but relatively high prediction accuracy in voxels surrounding hMT and extending toward pSTS. And so all of these voxels are potentially selective to bodies, but because we're using naturalistic stimuli, specifically two data sets that include motion as well, there's a good chance that our body features are correlated with certain types of motion. Think, for example, when you're walking, your arms tend to be swinging in a different way than your shoulders or your feet might. And so this raises the question of whether this is real selectivity to bodies. So in order to determine whether this is actually body selectivity, as well as to ascertain the degree of overlap between body and motion selectivity, we also extracted motion features from these same stimuli. To do this, we use spatiotemporal gabors that vary in location, size, spatial frequency, direction of motion, and speed of motion. So as you can see in this video, we captured a very detailed picture of the motion in our stimuli. We then fit two additional models, just like our body model, one using just motion features and one using both body and motion features. We computed prediction accuracy at each voxel for these models and squared these prediction accuracies to determine the amount of variance explained in each voxel by each of these three models. We then used a form of model comparison called variance partitioning, in which we subtract the variance explained by our individual models from the combined model in order to determine how much unique variance each of these two models contributes to our prediction of brain responses. Note that this also enables us to compute the amount of shared variance between these models, but we won't include these in the subsequent flat maps, though I have these in supplementary figures if you're interested in seeing them. So this is a visualization of the amount of unique variance explained by our body model and unique variance explained by our motion model in each voxel. If we look at regions like FFA and OFA, it's almost entirely orange, which corresponds to unique variance explained by our body model, but no unique variance explained by our motion model. In contrast, if we look toward the center of hMT, these are almost entirely blue, indicating a high amount of unique variance explained by our motion model, but very little by our body model. And if we look at this region surrounding hMT along this border here, we see a lot of white, which indicates unique variance explained by our body model and other unique variance explained by our motion model. So this confirms that these voxels are actually selective to bodies, but that they're also selective to motion independently. Finally, we wanted to see whether these body-selective voxels could actually be described as a few functionally distinct subregions. In order to do this, we used the weights that we fit in our voxel-wise model. And so if you'll recall, we have, for each voxel in the brain, weights relating how much that voxel cares about each of our body features. And so if we look at a voxel in FFA, for example, we see very high positive weights for faces, but very close to zero weights for other body parts. And so what we did is for each voxel in the brain, we averaged across visual field locations for each of our 6 body parts. So what this resulted in was 6 averaged values representing in general how much each voxel cares about each of our 6 body parts anywhere in the image. We then said which of these values is the highest, just thus telling us which body part each voxel seems to care about the most. That's what we have visualized here, where different colors correspond to different tuning to specific body parts more than other body parts. And so if we look at FFA and OFA, it's almost entirely pink, suggesting that almost all voxels in these regions are more selective to faces than other body parts. And if we look at this region around hMT, we see a mix of selectivity to a bunch of different body parts, but we can extract some general trends from this. Notably, if we look along the dorsal border, we tend to see a lot of selectivity to feet and some selectivity or to legs in red and to arms in dark blue. If we look along the rostral and caudal-most portions of this hMT region, we tend to see a lot of pink, which is selectivity to faces. And in contrast, if we look along the ventral border, we see a lot of green, which is selectivity to feet, and a lot of blue, which is selectivity to hands. This is generally consistent across participants. So here we have for subject 2, we see a lot of face selectivity, which is this pink, along the rostral and caudal portions of hMT and the surrounding areas. And if we look along the ventral border, we see a lot of green for feet and blue for hands. This is very distinct in our third participant, where especially on the left side, in the left hemisphere, we see tuning almost entirely more to hands and feet than other body parts. Finally, we also wanted to see whether these regions may vary in selectivity to different visual field regions. So we took the average weight for any body part in the left portion of the visual field and the average weight for any body part in the right portion of the visual field. And by subtracting one of these values from the other, we got a continuous metric of selectivity to the left or right portion of the visual field. And so, as generally expected, we see contralateral tuning in a lot of the visual system. And if we look at these voxels around hMT, we see relatively strong contralaterality along the dorsalmost border, but very weak to minimal evidence of contralateral tuning along the ventralmost border. We did the same analysis for upper versus lower portions of the visual field, finding that the dorsal portion of voxels surrounding hMT tend to be lower selective, whereas voxels along the dorsalmost border demonstrate more upper selectivity, which is consistent with retinotopy existing in hMT, but confirms that this holds true for the presence of body features as well. So to answer the questions that we raised at the beginning, we found evidence of body and body part selectivity in voxels surrounding hMT. We found a substantial degree of overlap between selectivity to bodies and selectivity to motion in voxels toward the periphery of hMT, whereas the center of hMT is almost entirely motion selective. And we found some evidence of distinct clusters of body selectivity in this region with selectivity to different body parts and visual field locations. So I'd like to say thank you to my PI, Mark Lesquois, to the fellow members of my lab and the funding that let me do this project, and to you for viewing. So, thank you.
Fantastic. Thank you, Matthew. And if you enjoyed the talk, please drop some praise in the chat in lieu of applause. So we have one question from Iris here on the panel. So she says, very cool that you can pull all of these selectivities from the naturalistic input. One thing I noticed was that hMT didn't seem more motion selective than say V1, V2, V3. Was that expected? And Iris, I will let you follow up as well. Just want to read these out for the folks watching on YouTube and elsewhere.
Yeah, so that's a great question. And this is one of our supplementary figures. This is the amount of unique variance explained by motion. And we raised the threshold so you can, since it kind of just washes out at the threshold we had before in order to show body selectivity. So in a way, this is kind of to be expected because we're capturing a lot, because we're using different spatial frequencies and different visual field positions in our motion model, we're capturing a lot about the low level, not kind of non-motion visual characteristics. And this makes it a little more clear as well that we see a lot more unique variance explained by our motion model toward the center of hMT than around here. So this did not surprise us exactly, but it is definitely an interesting finding.
Thanks.
Great. Do we have any other questions for Matthew before we close up the session? Okay.
Another question, if you.
we have a few minutes, like 1 minute.
Well, I'm just curious because so we're making quite a bit about that, so that you're seeing sort of on the edge of hMT, you're seeing selectivity, right? So what's your interpretation sort of functionally of that? What do you think? Why is that there? Do you want to say, are you trying to say that, you know, these areas are not as segregated as we tend to think based on the literature? There's more sort of diffuse selectivity, or that it actually replicates the literature really well?
This generally replicates the findings specifically of Weiner and Grossbechter's paper, where they found that body selectivity, they were trying to address this question of, are hMT and EBA overlapping or distinct? And they found this kind of crescent shape around hMT of body selectivity. And that's generally what we confirmed, though the exact shape of this crescent, they found it generally extended along the dorsalmost or ventralmost border, and we find it a little bit more dorsally distributed. As far as the reason for that, that's a great question, and I'm very curious to hear what other people think, because I'm not sure.
Cool.
And that sounds like a good place to end on, because unfortunately we're going to get kicked out of this room momentarily. So I'd like to thank all of our speakers once again for their fantastic talks. This was a really engaging session, and so thank you for being here. And reminder, meet with your Neuromatch matches and follow up with the speakers if you have more questions, if we didn't get to your question, or if you want to continue the discussion. So thanks again to everybody for being here, and also to our Neuromatch Backend technical host, Hadi Motamedi, for helping out run the session, and we will see you next time. Thanks, guys. Bye.